We're very excited to have Dr. Alba Grifoni back to the IEDB User Workshop virtual stage to present using the IEDB in your research, where Alba will take you through some of their lab's work and how they've also utilized the IEDB. Thank you so much, Alba.
Thank you so much, Nina, again for having me. And as I like to say, as at every workshop, I used to be an IDB user even before coming at LGI. So I've been a proud user of IDB since now 14 years. And so I hope that the talk that I'm going to give you today is going to spirit your curiosity in using IDB for your science. And so particularly the talk I'm going to focus today is about a novel approach toward a pan-viral family immunogen design. Essentially, so far we've been focusing on designing vaccine specific for different viruses. What if we try to find something that is in common between viruses of the same family and leverage that for a universal vaccine approach? And so because of that, I'm going to go over a couple of basic concepts because we need to rewire our mindset with that concept in mind. And so what do we think when we consider the concept of vaccine protection? And so that is a point that has been already raised during the pandemic. You could protect against actually having the infection. And in that case, antibodies, particularly neutralizing antibody and T-cell response, particularly in the mucosal side, are really the main actor that are able to prevent infection. One of the drawbacks of focusing against protecting against infection is the specificity. And so when we want to achieve this goal, a lot of time you need to focus on one virus at the time. Because as we've seen already throughout the pandemic, as soon as a variant emerges, you have a decrease of the neutralizing antibody response because the mutation actually impacts the interaction of these antibodies. However, if we consider in terms of protection from severe disease, then you can leverage the different component of the immune system that have an equivalent balance and contribution in actually slowing down the spread of the virus within the body. And so in that context, that's a potential for a pan-viral family approach. And that's because you took into account also the fact that single mutation may not necessarily fully disrupt the immune system. And so in that case, and particularly among the different population, my lab is interested in T cells. And the reason is because they have the capability to recognize multiple protein of the virus, which is an important feature, and I'm gonna highlight the reason why. So in general, we have been studied together with several brilliant scientists worldwide what is the contribution of the T-cell response in SARS-CoV-2 immunity. And that is kind of the contribution that now we are leveraging in the context of a pan-coronavirus approach. And so if we can essentially simplify what are the features that you should have, the first one is quantity, definitely a strong response and coordination with the other component of the immune system. They protect from severe disease in absence of vaccination. And that's a simplify of one of the study our group has been doing at the real beginning of the pandemic. The second component is kinetics. The faster the T-cell response are in recognizing and responding against the infection, the better is your disease outcome. The final component of that is the breadth. And so as I mentioned before, the ability of T cells to recognize multiple protein is associated with protection from severe disease. And so when we think about the ability of T cells to recognize SARS-CoV-2, another important feature is the ability to be quite resilient disregarding the variant of concern. And so whether you get a previous infection, a previous vaccination, or even if you experience an infection post-vaccination, your T cells are still there and are able to recognize no matter the variant of concern that you test. A final important feature is the fact that T cells do not get exhausted by previous infection and vaccination. And actually, if you experience an infection post-vaccination, a lot of time this infection is asymptomatic and is still able to boost the T cell response against non-spike protein primarily, but also is able to induce what we call de novo response that are variant specific. And so if we factor in that the T cells not only get boosted by the different vaccination and new infection, but they're also able to generate new response against the variant of concern, what we hypothesize in this bioinformatic pipeline is by factoring in these new responses, the T cells are able to bridge the gap between one variant and the other one. And so as it exemplifies right here, if we compare the T cell epitope repertoire based on the original SARS-CoV-2 Wuhan strain versus a much more recent variant, and you just look at 100% conserved epitopes, you go from 55% of the response fully conserved. But if you factor in the fact that the human population are usually exposed to closer SARS-CoV-2 variants, then this gap actually get reduced. And of course, as we've seen before, also in the context of the variants, our T cells are still able to recognize epitopes with a few amino acid mutation, and that is this gray here. So overall, that suggests that the T cell response are in general either very conserved or conserve enough that the T cells are able to cross-recognize the variants. So what I show you for now is that T cells can really recognize small variant differences, but they can do even more than that. And so at the real beginning of the pandemic, what we saw is that SARS-CoV-2 reactivity was detected in individuals pre-pandemic. And so this is what we show. Actually, 50% of these unexposed individuals already have a T cell response, and that was primarily driven by a response against the non-spike proteins, which are in general much more conserved. We linked that in a follow-up study to common cold corona, and this finding has been corroborated independently by other labs worldwide, actually pinpoints some relevant non-structural proteins or pinpointing specific region of the spike protein that are actually responsible to this pre-existing immunity. Doesn't matter, and so some of the study has started to stratify individual based on this pre-existing immunity at the beginning of the pandemic receiving a vaccination. And so as you can see here, essentially this pre-existing immunity provide a kickstart that induce a much stronger T cell response, which actually persists even a memory time point. And other studies have shown that pre-existing immunity to T cells are related to what are considered to be abortive infection. So individuals that get exposed to the infection, they don't get sick and they don't develop antibody against it. And that is primarily, once again, driven by non-structural protein. So essentially, all these different findings available in the literature led us to the conclusion that if we can broaden our T cell response to multiple antigen and potentially focus on what is conserved, we can develop upon viral vaccine. And so with this idea in mind, we developed this general scheme that can be applied not only to the coronavirus family, we're actually currently applying to different viral family. And so the first step, we do select a viral prototype per each viral family, and then we consider all proteins. And so that, as I said, is one of the beauty of the T cell response, which are virtually able to recognize any viral proteins. Then we leverage collaboration with the J. Craig Venter Institute to be able to compile a representative sequence that encompass the diversity of each family, which allowed us really to pinpoint what part of the protein is actually conserved across the family. And then on the other side, we instead look at the T cell response, and specifically per each protein, we really like to perform a fingerprinting, understanding what subregion of the protein are actually very important and recognized by the T cell response. And so we combine these two components together in what we call CEDARs, or conserved T cell epitope region, which then can be used to really understand if the general population already harbor pre-existing immunity that helps you recognizing other viruses, and also it could be used as a potential strategy to immunize people and focus the response so they're better prepared in recognizing future viral threat coming from the same family. Now, viral families are quite complex, and definitely each one of them have different diversity. So one of the first questions that we ask is, how far can we go when we consider this approach? And so in this study, we went back to pre-pandemic samples and defined the epitopes characteristic of alpha coronavirus and common cold beta coronaviruses. And then we asked the question, how conserved they are. And pretty much what we see is that alpha within alpha have the majority of conservation, and beta with beta have the majority of conservation. And so with that one, given that the beta coronavirus is being the subgenus of the family that has been the most evolving, we concluded that a pan-beta coronavirus approach is actually the most feasible. And so going back to our pipeline, the first question is, how are the immunodominant region recognized by the T cells? And here is where IDB actually take the spotlight, because thanks to IDB, instead of just leveraging experimental data done in our labs, we could leverage studies, which are almost 200 now in the context of SARS-CoV-2 epitopes in the context of humans, and really pinpoint per each of the different proteins what part is recognized by the T cells. So how do we do that? Pretty much as Professor Sette showed you at the beginning, we start by performing a query, and here I'm going to just show you an example in the context of the CD4, but by focusing on SARS-CoV-2 as an organism, T cell assay, class 2 restriction because we're interested in CD4 response, and the host human, we can then look at the different epitopes. Another point that we usually perform is to refine the query by using canonical size. And as Professor Sette mentioned, the database doesn't do that for you, but a lot of time if you want to look at specific epitopes, and particularly if you're interested in the restriction, narrowing down to the canonical size help you to clean up the data set available. And then finally, because we are interested in understanding what part of each protein is important, by using the antigen tab, we can go into the ImmunomeBrowser view, which you're going to hear later, but essentially what it's doing is compiling the different epitopes mapping on the same protein, so you really have a feeling of what is recognized and in which part. And the readout, it shows you both response frequency and epitope assay count, and they're gonna go over this later in the workshop, but we were able to essentially pinpoint per amino acid position what region are immunogenic, and we could download this information right here. And so with that information, we are having now a fully comprehensive understanding at the amino acid level of what is recognized by CD4 T cells. And so, of course, you can do exactly the same approach in the context of the CD8, and so that is how we were able to have a fingerprinting of immunogenicity for CD4 and CD8 T cell response. Now, the second component is to really understand how broad the conservation is without biases, and to do that, we leverage collaboration with the Craig Venter Institute, Professor Schuerman, which is now not in the institute any longer, and the ongoing collaboration with Professor Jim Tan and Christian Smeg really help us narrowing down this question. And so, essentially, what they are doing is accounting for the fact that you have a bias in the sequence available, particularly on SARS-CoV-2, and so they try to perform clustering analysis to select prototype, ensuring that diversity, no matter the sequence available, is maintained across the different subgenus. Thus, when we calculate conservation, we're not doing a pan-SARS-CoV-2 conservation, but we're actually taking into account the breadth of the beta coronaviruses. And so when we combine these two information, immunogenicity and conservation, this is what we see. And I'm starting with the spike protein, which actually is the least conserved, of many are familiar with. And indeed, the majority of our CEDAR are pretty much focalized in the S2 domain, and they really encompass a minor fraction of the entire protein. And so do we see a difference in cross-reactive potential on the T cells if we dissect our spike protein looking at our CEDARs versus non-CEDAR? And so this is work that my postdoc, Tertulliano, has been doing in the lab, and he essentially select prototype sequence based on the different subgenus, and then generate peptide pools encompassing the CEDAR and then non-CEDAR region, and then test for T cell immunogenicity based on looking at individuals that experience an infection. And the reason why we're going for an infection is, of course, because we're interested also in looking at the rest of the protein, and clearly we need individuals that we know they've been exposed to the entire virus. And so this is the data, and they are expressed as a fold change normalized by the SARS-CoV-2. So what we are seeing here is truly the extent of the cross-reactive response. Anything that is 1 and above means that the cross-reactivity is very high. Anything below shows a reduction of cross-reactivity. And so if we compare our non-CEDAR versus CEDAR, we do see a trend for increased response, particularly in the spike CEDAR. And it's significant when we look at viral species that the human population has not been exposed to. If we focus at the individual level, how many of these essential dots are still harboring cross-reactive potential, and we combine them together, we do see that at population level, single individual are cross-recognizing better if you focus the response on this CEDAR. For the CD8, the picture is exactly the same. We do see a trend. But overall, although we do see an increased ability of the CEDAR to enhance cross-reactivity of the T cells, the overall finding are a minor fraction. So as I mentioned before, one of the beauty of focusing on the T cells is that they are able to recognize multiple proteins of the virus, not just one expressed on the surface. So we went back and we performed our analysis again, this time considering the entire SARS-CoV-2 proteome. And what we found is there are mostly 3 different proteins that are quite conserved and immunogenic if we focus on the beta coronavirus. And if we just focus on Sarbeco, of course, there are other ORF-specific that add to the picture. And as you can see, depending on the protein, you could either have a very strong immunogenic and conserved type of selection. Pretty much the nucleoprotein in its entirety consists of that, or you can have additional different region if you focus in non-structural protein 12 and 13, which are related with the replication complex of the virus. And so what happened if we look at the single epitope level and we ask the question, are the T cells that are primed with the SARS-CoV-2 epitopes able to recognize any variant because they're conserved enough? And so to do that, we carry out a different type of experiments where we once again focus on PBMC coming from individuals that experienced an infection before. We expand them with the SARS-CoV-2 epitopes that we selected based on our analysis. And at the day 14 of expansion, we then test the equivalent sequence to ask whether we see cross-recognition. And so if we do this type of exercise in the context of the experimental layout, what you do see is if you expand your T cells, the response is going to be quite strong no matter how low you go in concentration. If you look at the variant-specific response, we do expect a reduction in magnitude, but that's still quite the cross-recognition, which is preserved in some cases even a lower concentration. When we focus on ORF, and this is very much about the selection that we did to account for additional antigen interesting in the Sarbeco genus, what we saw is that essentially no matter the variant that we tested, we have full cross-reactive potential. What finding that we consider interesting is when we went into the nucleoprotein, although the general protein has been selected to be immunogenic and conserve across beta coronavirus, at the single epitope level, this cross-reactive potential is really different. And you may see some epitopes that are much more recognized toward the common cold corona and others more recognized toward the Merbeco. And so this once again suggests that instead of using epitope-based approach, if we expand our region, considering multiple epitopes, that reestablish again the capability overall of the T cells to recognize any viral species included in the beta coronaviruses. So to test that hypothesis, we went back on our ex vivo assay, and in this case, we decided to compare the full spike protein, which is currently given in the majority of the COVID vaccination, compared to our rest of the proteome CEDARs. And then we test that again using the same layout, same type of donors, and using the same type of analysis. And so what we see now is quite different and striking. Now there is a significantly higher difference if we focus on the full spike protein and our rest of the proteome CEDARs, which is significantly higher, even in the context of viral species that we haven't been exposed to, and also significantly higher also at the individual level. And that is also true whether you look at CD8. So pretty much the approach that we select shows enhanced cross-reactivity in both T-cell population. Now, if our goal is to focus on this conserved region, the other component is what is the population coverage? Can we utilize this approach disregarding geographical location? And so to ask this question, once again, we went back to the IEDB, and in this case, the analysis tool, and we designed, thanks to the help of Dr. Rafael Trevisani, a customized pipelines that combine T-cell prediction with the population coverage tool you're gonna hear later in the workshop. And essentially what we do see is that although the coverage for the full spike protein is already pretty good, and unsurprisingly, given the COVID vaccine are actually working in protecting from disease severity, we do see an enhanced population coverage if we incorporate the ARP CEDARs. And particularly if you focus on the worldwide coverage, the increase is roughly twofold, really suggesting that the approach that we have applied not only increased in terms of magnitude, but also has the potential to increase population coverage and maybe reach specific individual or population which didn't have enough predicted epitopes on the spike protein. And so to conclude the talk, we have developed a pipeline that is able to identify, conserve immunogenic region in each viral family, which could be used as a general pipeline to apply to different viruses. Our CEDAR approach has demonstrated that T cells prime with one virus have the harboring the potential of cross-recognized diverse beta coronaviruses. And in specific, if we include the rest of the proteome compared to the spike, what we see is that we significantly expand the breadth of the T-cell response and also expand the cross-reactive potential. That also gives an improvement in terms of global population coverage. And so this work really shows feasibility of applying this selection to other viral families. Now, as a next step of this type of pipeline, we are asking additional questions, which I'm not going to have the time to explore today. But one of the questions is, although we do have a very narrow CEDAR selection, are we sure that actually constant vaccination is not still bringing us toward a minimum amount of cross-reactive T cells able to recognize different viruses? And so that is a question that one of the members of my lab is doing. The second question is, can we actually package these different CEDARs and use them as an immunogen? And so that is a completely larger line of research that we're currently exploring by combining AI prediction to link the different CEDARs and testing in animal models. We're going to be able to address that question. And so far we have promising results. And so with that, usually I start thanking my team, but I think in this specific case, I would like to start thanking IEDB. All the work that I've shown you so far has been using IEDB database and tools and was really not possible without the existence of IEDB, and really showing you how this curated database is really allowed us to bring to the next level the experimental work that each one of us is doing in his own different lab worldwide. And so that is really a very precious feature, and I really encourage all of you to use it with the same enthusiasm that I entered in 14 years ago. But with that, I would also like to thank my team, and particularly the work that I show you today has been carried out by my postdoc, Tertulliano, that now is currently pursuing a different postdoc in WashU. The follow-up work that I briefly mentioned, asking what's happening on the different vaccination with spike, and are we already there yet in cross-reactive potential, has been currently doing by my postdoc Zi Wang, as well as the question about the model testing. And finally, as I mentioned before, also we're trying to apply this pipeline to different viral families. And so Mars Kim in my lab is applying that into the context of enterovirus together with Naomi, and Matthew Weisim is focusing on the pox. Then I would like to thank all the collaborators, Alessandro Sette, the Craig Venter Institute, and other collaborators on the funding. And so with that one, I will take any question. Thank you.
That's awesome. Thank you so much, Alba, for that wonderful presentation. It's so great to see Alba, who's presented over many, many years and is always presenting the most cutting edge research to show everyone not only how you can do really great research, but how you can apply the IEDB to that research as well. So thanks once again. And it's great to see the lab working on some really cool things. We are running about 10 minutes ahead of schedule, which means I can give Alex and Alba a little bit more time to check the Q&A and to start thinking about which questions they may answer. It also means you, the attendees, do have some time to think of a few more questions and post them while I give a couple of updates and reminders. So the start of the workshop, and actually, sorry to all, if you do have a question, please do post it in the Q&A rather than the chat. It just helps our presenters really manage them and make sure we answer all your questions as well. So yes, please do post in the Q&A. But yeah, so the opening of day one, as you can see, is an introduction from Alex and Alba, where Alex touches on all parts of the database, and then we jump straight into research examples and how you can use it. So that gives you a big picture overview from the get-go, and then we spend the rest of the session really diving into different aspects of the database and the resources available. And as Alba mentioned, the population coverage tool, that's something Alex will discuss later on this afternoon as well, or later on this morning. But with that, if it's okay, Alex and Alba, I would just very quickly take the first question. We had a question come through to say, hello, do we get the audience at any point to be able to turn on their cameras in this session, which would be really lovely. But as a reminder, we've had over 630 people register for the workshop, and we are in a Zoom webinar format. So it may get tricky having people turn on cameras. So the webinar format restricts a camera and sound for the attendees, but we thank you for your understanding. It is just to help us manage everything really efficiently and nicely. But the exciting part is that we do get so many attendees. So again, thank you. And then now introducing back Alex and Alba to please start the official Q&A for section one. Alex, feel free to take the first question.
Right. So the first question from Scamp is, what means an epitope with high rank and low score? So I'm going to deflect that question because it's a question that relates to the analysis tools, the prediction tools, which is going to be discussed later. But as a preview, it's not uncommon to have a low score. If a low score refers to this is the 1%. So often a low score means good, but other times the scores that were high numbers means good. So Bjorn will cover that later on. Alba, let me know if you want to take any questions. Otherwise, I'll keep going through.
I select one, but go ahead.
Go, you go.
It's down on the list. So Laura Gibson, question for Dr. Grifoni. How do you decide which epitopes in IDB are immunodominant? Example from ImmunomeBrowser peaks, when the data depends on individuals choosing to submit their data. Seems a large selection bias. Thank you. Thank you, Laura. That is a very good question. So let me just clarify a couple of points. First of all, it's not up to the individual necessarily just to submit their data, and there's going to be much more into this workshop where the curation system essentially guide you through how they read the papers and actually curate the epitopes. So that is a little bit removing the bias of the single individual. Do I feel like submitting the database or not? Actually, there is much more behind it. The second point about the selection bias on the antigens, yes, absolutely. One thing that we usually do when we compare the IDB database is also to compare with our own data. And so in 2021, we performed a large-scale epitope identification study with over 100 different individuals where we really narrow positive and negative responses. And so we usually use this type of data set that are curated in our lab, comparing with the IDB to ensure that bias is not encountered. And a lot of time what happens is simply that the IDB data, if anything, they threaten some immunodominant region that we already see in our data. And so it kind of makes us more confident in our selection. And I leave that to Alex, the next one.
So how can the IDB be used in developing anti-snake venom detection and venomics? So this is a very specific question, but I would like to use it to kind of broaden the field. How can the IDB be used in developing X ... or X? In general, what we recommend is that first you might look at the database to see if there is any data related to a particular protein. For example, if you have one particular venom in mind, maybe someone has already studied that venom, has already defined what are important epitopes for T cells or B cells in humans or in mice and whatever. And that would be always the first step. Then the next step would be if you have your venom that you want to develop an antibody against, you may run antibody epitope prediction tools, or if you want to develop some strategy that involves T cells, you may run T cell prediction tools, and that again, you may there are tools that predict the immunogenicity for the human population or in different animal species that might be used to test. But in general, the process should be look in the database first and do predictions after that.
All right. I would like to thank Mauricio. I'm going to read both questions together. Is there a correlation between antibody levels and response to conserved CD4 T cell epitopes? And does the antibody response target conserved region containing T cell epitopes? Thank you. That's a very good question. So we haven't looked into whether there is a correlation between the antibody levels and the response to conserved CD4 T cell epitopes in this study. However, as I mentioned before, the lab is currently looking at a spike-based vaccination to answer that question. So we're going to be able to look into that further. In terms of does the antibody response target conserved region containing T cell epitopes, and so you can leverage the IDB that contains some of this information as well as the literature. So as you know, we need to think about the quality of the antibody response. And so classically, neutralizing antibody are against the RBD region. However, there are several non-neutralizing antibody and specifically a couple of neutralizing antibody in the S2 region that actually coincide with our CEDAR region. So our expectation is that there is some overlap.
So an anonymous attendee says, to my understanding, epitopes are peptides that trigger immune response. So if I'm looking for peptide in the MHC assay tab, not the T cell, would these peptides still be considered epitopes? Very good question that goes back to general phenomenon or a general rule that in order for a peptide to be a T cell epitope, it must bind MHC, but binding to MHC is a requirement, but it's not sufficient. So you can have things that bind but are not epitopes. And for example, this could be a situation in the case of a self-peptide that might bind very well, but if tolerance has done its job, it might not be recognized from T cells. Further, that allows me also to further underline the key thing of the IDB, which is the IDB curates assays. And so the fact that that particular peptide was found to bind to MHC, but say was not found to be recognized in the context of someone with chronic hepatitis, well, maybe someone with acute hepatitis may recognize that epitope. So what is an epitope or not, to say something is an epitope or not, it's perhaps a faulty way to look at it, because you have to tell me which biological context are we talking about. So again, it could be that someone that has been immunized with a vaccine has a completely different response to someone that instead is having a chronic infection, or someone that is immunocompromised for other reasons because, say, is undergoing cancer treatment, may have a different response of someone that is not.
Well, really insightful presentation, thank you. Is there any possibility that expanding the antigenic breadth of these T cells could pressure the viruses toward mutation in this conserved region? And are there any risk of autoimmunity, self-reactive T cells in the context of this work? Thank you so much for the positive comments, and I love the question. It allows me to add some of the work we have cooking in the lab. So the first point about the possibility that you can increase pressure, hypothetically, we don't think that would be the case, considering that already the human population are exposed to these different viruses, and particularly some of the targets that we're looking at it are related to the polymerase reaction, which means that mutation in the wrong position might actually impact viral replication. So the likelihood is less than what we think, considering instead surface protein that have different evolutionary pressure, first in terms of viral entry and also antibody responses. I love to dive more into the question about autoimmunity, because we actually develop a pipeline to ensure that is not the case. And so particularly when we try to combine the different CEDARs in a immunogen-like design, we made a point to actually add an additional bioinformatic analysis to make sure that not only the epitopes we select, but also potentially new epitopes generated by our linker region are not high homologous with self. And so by adding this additional pipeline, we're able to modify the linkers so that even when we design a construct, the sequence identity with potentially self-antigen is very low, and so we should not have that risk. Thank you.
So there is a question: how much data is available for non-conventional animal models such as pig and cattle? Short answer is not not a lot. The longer answer is not our fault in the sense that the IDB curates everything that is out there. And I'm with you that certainly the other non-conventional animals are perhaps understudied, and it would be good to have more epitope definition and MHC definition in those species. The lion's share of what we find in the literature and we're able to curate is human, then is mouse, then is non-human primates and other things. I specifically looked while we were talking, and I ran a query for how many cattle basically Bos Taurus, domestic cattle. Epitopes are there, and there's about 2,000 epitopes compared to what we had. I forgot the total number of epitopes was 2 million structures. So it's a very small fraction of the data that is associated with cattle, and I would imagine that the same would be true for pigs.
I have a question about awesome presentation, Alba. Shirin Soleimani. I hope I didn't butcher your last name. Thank you. Have you also thought about CEDARs in the field of cancer antigen, and do you know any ongoing project focus on that? So the approach that we take on the CEDARs may not necessarily be applicable in the context of cancer, where the idea is going outside, something is conserved in principle and just focusing on neoantigen. Then if your question is, can we kind of look into common neoantigen, that is a potential approach that we haven't considered, but we could in the future. As far as I know, I don't know any ongoing projects that are doing that. Thank you.
So is the server applicable in parasite immunology? Yes. Quite a few of the records in the IDB are derived from parasites, trypanosoma, malaria, and so forth. And if it's out there, we curate it. Also, certainly the predictive tools and analytical tools or predictive tools that Bjorn is going to discuss have been applied to predict and identify epitopes, for example, from Plasmodium and in the malaria context. And then I will take another question that came up from Gonzalo. Sorry if it was mentioned and I missed it, but there is a system in place for users to submit their epitope data for curation and potential incorporation in the database before and after peer review publication. So the answer is yes, to a degree, and this historically is linked to the fact that the NIAID has supported a number of large-scale epitope identification contracts in infectious diseases, in allergy, in autoimmunity, and the people receiving that funding were actually committing to submitting their data to the IDB. And in fact, a significant fraction of the data in the IDB is derived from these direct submissions. The direct submissions also have an advantage that we encourage in the direct submissions people to submit negative data, which is very important for what I was alluding to earlier. That is great to know. It's something that is not in the database. It's not in the database because it was tested and found to be negative or was never tested. And so that is very useful data, but people don't usually publish, hey, this is a list of 10,000 things that I tested and they're all negative. After saying that, whether we accept direct submission is more on a case-by-case data because we would like to have some degree of essentially assurance on the quality of the data, in the sense that the IDB primarily curates things that are peer-reviewed and so have passed some degree of scrutiny. But on occasions, we have accepted and curated direct submissions from users.
So a question from Aviral Kaushik. What type of vaccine does IDB help us to design, DNA or mRNA based? If both, then is there a difference in the pipeline? Thank you for the question, because it allows me to clarify. The IDB is not able to inform us on vaccine platform. It actually just provides us information on what antigen or what antigenic portion is relevant in the context of the immune response. And in fact, the lab is currently considering potentially different vaccine platform to see which one is actually the most indicated depending on the viral family. And that work is unfortunately still not available as a prediction pipeline in IDP. So I think more experimental work is needed. Thank you.
Question, what is for you an important point that you could exclude a paper before integration in the IDB? We have some very well-defined criteria for what is the minimal type of information that needs to be associated with a record for us to be able to curate it. And those are spelled out in the curation manual that actually is available online. But essentially, in order for us to curate a record, there must be a structure. I mean, we can't curate an epitope if you say, well, I tested some peptide from hepatitis. I mean, that doesn't mean anything. Or you say, I tested a peptide residues 20 to 35 of surface antigen. Well, that also doesn't tell me anything because I don't know what sequence you used and regarding whether the pre-S sequence is included or not. Residues 20 to 35 is a completely different structure. So there must be a defined structure. And also, for example, there must be some degree of narrowing to a structure. Again, if you say, I have an antibody that binds to Mycobacteria tuberculosis, well, that doesn't tell me anything because there are 3,000 different ORFs in Mycobacteria tuberculosis. So it has to be mapped to say, a 50 amino acid residues. Another fundamental criteria is that there must be an assay. So you can't say this epitope was positive for immunogenicity. Well, again, that doesn't tell me anything. You have to tell me, I tested this for interferon gamma release using an ELISpot assay. And finally, where is this happening? Was this done in cattle, in pigs, in humans, in mice? So there must be some degree of meeting minimal criteria for inclusion. But beyond that, we curate everything.
Excellent. Thank you so much, Alex and Alba. We are at time, a couple of minutes over, and I know still more questions. So I can see some of the team is already responding via a typed response. So are there any last questions or comments, Alba or Alex, that you'd like to make before we move on?
No, you make us work too hard.
I know, it was a marathon.
Can I say again, I love IDB, and that's it. Bye.
Thank you so much, Alba. It's wonderful as always to have you, and thank you, Alex. And as I mentioned, there are still a few questions, so our team will do our best to type a response if we haven't answered them live. And so there should be an answered column in your Q&A function, and then you'll see text under the questions that we've responded to via typed response.